Hey guys, welcome back to Sci. In this lesson, we're going to be talking about the nucleus, endoplasmic reticulum, and ribosomes. Now, in the last lesson, we talked about the cell membrane, and if you want to review that, check the last video in this playlist. Now, before we dive deep into the structure of the nucleus, let's talk about the function. The nucleus holds within it DNA, or deoxyribonucleic acid, the genetic material of the cell. And this codes for things like metabolism, reproduction, protein synthesis, and nearly every function of the cell. So it's extremely important to protect the genetic material. And that's where the nucleus comes into play. Now the nucleus is made up of two membranes. This is called the nuclear envelope. And within the nuclear envelope, you have this perinuclear space. You also have holes that make up the outside of the nuclear envelope, and these are called nuclear pores, and these regulate which molecules come in and out of the nucleus and which molecules can access the DNA within. And so these can be regulatory molecules, these can be RNA exiting the nucleus, and it's just extremely important to regulate what is accessing the DNA because it's imperative that it is protected. Now that we've gone over the outside layer of this nucleus, within the nucleus you also have a structure called the nucleolus. And the nucleolus does not actually have a function that's correlated with chromosomes, but is instead involved with the production and the creation of ribosomal RNA, which can be used to produce ribosomes, which we'll be talking about later in this lesson. Now, the nucleus can have multiple of these nucleolus, and these are called nucleoli, and that's the plural form. So nucleoli. But it's extremely important to understand that within the nucleus, there's not only the DNA, but also this nucleolus. Now continuing on, the rest of the nucleus contains the DNA. And the DNA within eukaryotic cells can be up to 5 or 6 feet long. And so it's extremely important that the cell has a method of packaging the DNA within this microscopic nucleus. And the way this is done is using histone molecules. And so here I've drawn an example, and histone molecules are positively charged. Let's draw that out better. Positively charged, while the DNA that is wrapped around it is negatively charged. And so because of this, the DNA is attracted to the histone molecule, and so it wraps really tightly around it. Now, this can also be used in gene expression, where DNA methylation or adding methyl groups to the DNA can cause the DNA to wrap even more tightly around these histone molecules, and therefore it can control gene expression and kind of lower certain genes that are not necessary for certain cells. Now, continuing on, it's important to understand how these structures correlate with each other. You have these chains of histone molecules that make up nucleosomes, and these further can create chromatin fibers. And during cell replication, these chromatin fibers come together to create chromosomes. We'll be talking about what these chromosomes do or what their function is later on, but it's important to understand how the cell is packaging the DNA because it's an incredible feat in and of itself. Now that we've talked about the nucleus, you can notice that there's this folded structure that is on the outside of the nuclear envelope, and it's connected to these nuclear pores. And now what this is called is the endoplasmic reticulum. And so you have two parts of this endoplasmic reticulum. You have the smooth and the rough endoplasmic reticulum. And the way that you understand these two different divisions of the same structure is that the rough ER, or endoplasmic reticulum, has ribosomes on the outside. And so here we have ribosomes on this darker blue structure that is the rough ER. Now there's also two different functions for the smooth and rough ER. So the smooth ER is involved in lipid production and lipid creation. So cholesterol, steroids that are used in signaling, these are all produced by the smooth ER. You also have control of toxins, and so it detoxifies certain things that are entering the cell, and your liver cells actually have a larger amount of this smooth ER because the liver is involved in detoxifying food that we've consumed. Next, you also have the rough ER, and the rough ER again has these ribosomes on the outside, And so it's involved in the regulation of the proteins that are synthesized, the RNA that is going to these ribosomes, and also where these proteins are moving to after they are synthesized by the ribosomes, because they need to be further edited and also moved to other parts of the cell, such as the Golgi apparatus or the cell membrane. And so that's what the rough and smooth ER are involved in. Now the ribosomes themselves also have specific characteristics. And so you have a large subunit and a small subunit. And the way that we look at this is you have mRNA or messenger RNA that goes through these subunits, and codons within this mRNA attached to tRNA that has specific amino acids that correlate with these codons, and they're attached together to create polypeptides. And I'm going to be going into much more detail about this process. This is just a basic overview of what a ribosome does, but we'll be talking more about this when we talk about protein synthesis. For this lesson, we're specifically going to be talking about the structure of this ribosome. And as you can see here, we have 60S and a 40S piece of these ribosomes. And these correlate to the large and small subunits that make this up. Now, these subunits are made up of rRNA or ribosomal RNA that is produced in the nucleolus of the cell, and you also have ribosomal protein. Now, these two parts, the 60S and 40S parts, are dictated based on the densities of these structures. So the S correlates to the Svedberg unit, and it's a unit for sediment rate, and it's controlled based on the different molecules of rRNA and proteins as well that make up these subunits. And because these molecules have different masses, densities, molecular shapes, you have these S values for these molecules. And so the large subunit, again, for eukaryotic cells, has a 60S value, and the small subunit has a 40S value. Now, in prokaryotic cells, you have a 50S, and you have a 30S value for these large and small subunits. So in total for this eukaryotic ribosome, you have an 80S value. And this is just based on the sediment rate of the overall ribosome, and so those 60 and 40 don't add up to 80. Just a basic understanding is that these subunits, when combined, have this 80S value. And if you want me to go much further into ribosomes or other proteins and how these values are created, just let me know in the comments below, and I can go into much more detail, but we're just giving a basic overview of the structure of the ribosome, and so this is just helpful for you to understand what a ribosome is. Now, to summarize this full lesson, you have the nucleus, which is made up of the nuclear envelope. You also have the nucleolus, or multiple nucleoli within it that produce ribosomal RNA, and you also have the DNA that is bunched into chromatin fiber, and this is through histone molecules, and attached to the outside of this nucleus is the endoplasmic reticulum, or the ER, and you have the rough ER that is involved in protein synthesis and has ribosomes attached to it, while you also have the smooth ER that is involved in lipid production and also the detoxification of toxins. And finally, the ribosomes that cover the rough ER and also can be free floating within the cytoplasm is made up of a large and small subunit. There are millions of these ribosomes within the cytoplasm, and they are used in protein synthesis to produce these polypeptide chains that are later combined to create proteins. Well, thank you for listening to this lesson. I hope this was helpful. If you have any questions, please let me know in the comments below, or if you have any information that you want me to go over, I can also do that. In the next lesson, we're going to be talking about the Golgi apparatus and other organelles within the cell. Thank you.